To finish this chapter, I'm going to start discussing the cytoplasmic male sterility. As I said before, cytoplasmic male sterility means that the direct influence is coming from the cytoplasm while there is modifying genes in the chromosomes that modify the process. If we have sterile cytoplasm, we call it S or CMS, and it produces non-functional anthers or pollen, while the normal cytoplasm usually produce functional pollens. The genes that modify this sterility, we call it fertility restoring genes, and we refer to it as RF. It is a dominant gene, and in the presence of these genes, the sterile cytoplasm background produce fertile ones. For example, if we have S cytoplasm with RF capital, or S with RF capital, RF small, it produce male fertile. In the case we have normal cytoplasm, whatever alleles we have or genes we have, it produce fertile only. So with normal cytoplasm, RF capital, RF small, or RF capital, RF small, and RF small all produce fertile. The only one which produce sterile plant is the one with the sterile cytoplasm or S cytoplasm and RF small. Is how the sterile cytoplasm produce. Usually it's produced by inducing or introducing of foreign nucleus into a cytoplasm. And because it is cytoplasmic effect, it is only transferred by mother plants. And this figure here shows you how this transfer. If we have CMS with RF small in the ovule, and normal or CMS with RF capital, then the pollen will be RF capital. So it will transfer to the CMS RF small, and when they cross produce offspring, it will have CMS which coming from the egg, but the restorer gene that came from the pollen, RF capital, RF small, produce a normal pollen or normal gametes. That's how the even though it is coming from, that's how the CMS is coming from the cytoplasm of the plant. So it is transferred by the mother. Now, if this F1 hybrid is crossed with CMS RF small, then it will produce 50% RF capital, RF small, and 50% RF small. With CMS, it will produce half sterile, half non-sterile plants. Now let us assume that we have one male fertile restoring genes, and what are the expected progenies to be produced if we cross it with different pollinators. For example, if we have S RF small, and S RF small, the same mother, and we cross it with S RF capital, we are expecting to have heterozygous RF capital, RF small, all fertile. While if the pollen source is heterozygous, we are expecting of RF, where we have RF capital RF small, we are expecting to get 50% male fertile and 50% male sterile. If we cross it with normal but RF small, we are expecting to get all of it sterile cytoplasm with RF small and all male sterile. It is like any normal process. The second question that we want to answer now is how we can utilize and when the breeders utilize cytoplasmic male sterility. Most of the uses of cytoplasmic male sterility was found on producing of hybrid seeds, and most of the crops that they used for cytoplasmic male sterility was corn and sorghum to produce hybrids of these plants. The other options that breeders were using, in addition to genetic and cytoplasmic male sterility in hybrid production, was what we call it gameticides or pollen suppressants or chemical hybridization agents, which is chemicals that found to induce male sterility, and it has been used to control the pollens during the commercial hybrid seed production, and in this case we don't need to do the emasculation process. Most of the time these chemicals were sprayed on flowering period before the or sometimes before flowering in order to inhibit the production of viable. Even though this is a good materials that has been used, these chemicals, but it has big problem because sometimes it is not produce complete pollen sterility due to many factors. First of all, not all the genotypes respond the same. Second, sometimes certain environmental conditions are not suited to produce completely. They are leading to production of some fertile pollens. The other thing is the chemical itself. Some of the chemicals are not reaching the floral organs because they are not translocated in the plants. They are not absorbed very well in the plants, or they are spraying not on the proper time, or the needed dosage is not sprayed. So there is big problems that facing us in using chemicals. If these chemicals has to be used, we have to first of all standardize to make sure that this genotype is respond very well, to standardize the environmental conditions around it during the spraying, and that this chemical is translocated to floral organs, and we have to spray it on the proper time and using the proper dosage to inhibit all the pollens under different environmental conditions. Otherwise, it will not be useful. Another problem that not all the plants are producing determinate flowering, which means that not all the plant flower on the same time. We have some what we call it indeterminate flowering, where it flower this time, but it is end with pollen, bud, and not flower, where the bud is growing and producing more flowers. So when we want to spray, we have to spray on many times, and maybe the cost of spraying will not be as as the determined flowering, and in the same time maybe some of the flowers will produce normal pollens when it is developed. The advantage of these chemicals that there is no need to control and maintain cytoplasmic male sterile or fertility restoring parent lines, or we should not look for any sterility genes and introduce it in the species, in the plants, and maintain it for a long time. So it has some advantage, but there is some disadvantage. Therefore, it is the decision of the breeder to decide which method to use for male sterility. As you see, and you know, in the beginning of this chapter, one of the main uses of the fertility or infertility was such as incompatibility or male sterility or cytoplasmic male sterility, is that we are promoting cross-pollination and reducing selfing. Unfortunately, you know, as you know from chapter 2, we have sometimes some seeds that produce asexually. These seeds that produce asexually through apomixis are one of the barriers that prevent cross-fertilization. And some of the plant species are called obligate apomictic, which means that they are not produce any seed except by apomictic seeds. And this case, how we want to do recombination and introduce new genes. This is one of the problems. Some of them, they are called facultative apomixis, and in this case it can produce sexual and apomictic embryos, and it can occur in the same ovule of the plants, such as a blue grass. In this case, we have to separate both from each other in order to increase the recombination and the outcrossing of the species, in order to introduce new genes to improve this. If you remember in the beginning of this chapter, in part 1, when I was talking about the importance of producing seeds during crosses, I told you this is more important if we are doing interspecific or intergeneric hybridization, because these type of crosses usually doesn't produce that much seeds, and the seed production or the viable seed production is dependent on how related are these species to each other. Therefore, I'm going to discuss some of the methods that we use in order to make sure that the seeds are produced in case of interspecific hybridization, and the methods that were used by breeders to produce seeds. Some people will ask if this is a problem, interspecific or intergeneric hybridization, in doing this and producing seeds, why we are doing that? The reason that we are doing that is that sometimes we have a problem that we cannot find in our cultivated plants or the plants that can cross with it any genes. Therefore, we are going to some other species or genera that has some relation with our crop and it can cross with it. And in this case, we need to have this type of cross to transfer these genes. And we cannot produce bring any gene from any place with our conventional crossing or hybridization breeding. This is available because of the biotechnology, but before that it was not available. Therefore, we have to make sure that the two species can cross with each other and they can produce good seeds. And this can be easily if we study the morphology and the physiological characteristics of both species, and we make sure that these two species has certain relationship and they can produce good amphiploid and F1 seeds that can survive and be a successful cross. And we have many examples of the related species that has been used in the past for producing hybrids, or they have good cross fertility among these, which is in the second slide here, where we have, for example, glycine max with glycine soja, which is a wild soya bean, and glycine max is the cultivated. In cotton, the American upland cotton with the American Pima cotton, and in Zea maize, which is the Indian corn or maize, with the Zea mexicana, and all of the, or the Teosinte, all of these were very successful crosses between two different species. The previous examples are direct way of doing crosses, but sometimes if we cross certain species such as the autopolyploids, they cannot survive by themselves. For example, if we bring two diploid species, cross them, they will not survive, or two autopolyploid and cross them, they will not survive. So what we are doing, usually, we cross the diploid species and the F1 plants were subjected to colchicine, and we double the chromosome of these plants and we produce F1 of tetraploid that can survive. And these examples were found in many fertile tetraploids in Brassica, Triticum, in Nicotiana, and other genera. So it cannot survive as diploid, but when you double its chromosomes, it will survive and give us a good new species of autotetraploid that can survive and produce seeds, and we have many examples. Another way to do that is what we call bridge breeding. A bridge breeding, when we have, for example, cultivated tetraploid plants and we want to transfer gene from a diploid related species. In this case, if the plant that we are we want to get the gene from is diploid and our plant is tetraploid, what we do is that we bring the diploid species, double its chromosome, so it will be tetraploid, exactly like the plant that we use it for cultivation. And then we cross two tetraploid species with each other, and we transfer the gene that we need. In this case, sometimes we are transferring the genes that we want and that we don't want. So in this case, we can back across the new hybrid we brought from the wild and our cultivated crop to our cultivated crop many back crosses and then get a exactly like the cultivated plus the chromosome or the gene that has the gene that we need. Or sometimes we do it in reverse way. Instead of doubling the diploid species chromosome, we sometimes do a haploid of tetraploid. So we do a haploid of tetraploid, then we cross it to the diploid species, and the plant that produce, we can redouble it another time, the F1, and produce what we call a new hybrid species with all the genes that we need. So either we double the genome of the diploid and cross to tetraploid, or we produce haploid from tetraploid, which we call it dihaploid, and cross it with the diploid, transfer the gene, and then redouble it, double the cross of, chromosome of the F1 again, and produce tetraploid cultivated plants. And both of them has been used. For example, in potato, Solanum tuberosum, they are using this process, which is dihaploid with diploid, and then redouble the chromosomes to get a new autotetraploid species. By this, I think we finish this chapter, and we hope to start with chapter 8 soon.